Early on in the semester, when we started talking about Mendelian genetics, many of you had questions for me, such as, well, we understand that in plants, pea plants, we can have tall plants and short plants, and this is a trait that's governed by a single gene and two alleles, and all that made sense. But what about people? We don't have tall people and short people. We have people of all different heights. And the same went with eye color and hair color. Many of you had very good questions for me about these traits as they related to your own family, your parents, your siblings, and also as they related to people in general. And I kind of put those questions off all the time, and I said, we're going to do this at the very end of the semester. We're going to talk about human traits and how humans look blended, even though blending is not true. It doesn't adhere to Mendelian principles. And so finally now the time has come. This lecture on Chapter 24 from your textbook on quantitative genetics will address those issues. How do humans look the way they do, like blended averages between their parents, when in fact the blending theory is not accurate? So today we're going to start off just by defining quantitative characteristics. What are these? What do we mean by that term? And how does quantitative genetics play into that? Part of that definition will have us talk quite a bit about polygenic inheritance. We've already talked about, in general, traits having multiple gene inputs. So it's not one gene governing one trait, but multiple genes governing a trait. But we'll kind of expand on that and give some meat to that skeleton. We'll define a term of heritability as it applies to quantitative genetics. Not just the idea that traits are inherited, but heritability means something in the quantitative genetics field. We'll discuss the differences in variance between genotypic and phenotypic variances in this idea of heritability. And finally, we'll define where the limitations of this concept of heritability lie and what can and can't be inferred when one studies heritability. But all that will become clear as we go through the lecture. So we have briefly covered the basics of this chapter earlier in the course. We'd said that not all traits are one state or another, in other words, tall, short, green, yellow, round, wrinkled, even human traits like having cancer and not having cancer. In pea plants, remember we made the point that Mendel was very lucky. He happened to choose these traits that had two states, one or the other, and had only two alleles, and that allowed him to kind of set his laws of dominance. But not all traits are that way. In fact, the majority of traits do not behave this way. So yes, in pea plants, we have tall plants and short plants, and that's it. We have no in-the-middle plants for pea plants. We have no medium-height plants. It's either tall or short. Traits that behave this way, where there are two states and only two states, are referred to as qualitative characteristics. Qualitative meaning it doesn't need to be measured. You don't have to measure the height of the tall plant to tell that it's tall, and you don't have to measure the height of the short plant, in this case, to tell that it's short. You merely have to look at them, and you can instantly determine qualitatively which characteristic the plant has, tall or short. Most qualitative traits, although there are few of them, are governed by Mendelian rules. They're governed by a single gene that has two alleles, kind of classic complete dominance cases. But most traits in most life forms are much more complex, especially in higher eukaryotes, such as mammals. Traits in higher eukaryotes, and in other organisms too, express phenotypes across a very wide spectrum. So you might have very, 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 very tall and very, 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 very short, but you have all of the different heights in between, as shown here. So you have this spectrum of traits where there are two extremes on either side, but many, many organisms in the middle at the average, and organisms hitting or having all heights in between those two extremes. Because of this, we can't just look at the plant and say it's tall or it's short, because we don't have two states, for one. And two, because since we have this varying across the spectrum, we have plants of all different heights, so we can't just look at one plant and say that it's tall or short. We must measure these traits. We can't say that the plant is tall. We have to say that the plant is 11.5 inches tall. We can't say the plant is short. We have to say that the plant is 3.75 inches in height, making it short. So these types of traits require measurements. We refer to these then as quantitative characteristics because we have to attach numbers to them. They need to be measured. They are quantitative. And most quantitative traits, they make up the majority of most traits in most life. Most quantitative traits are governed by many, many different genes at many, many different loci or different areas around the genome. And in addition, there are often more than 2 alleles per gene in this case. Complicating things even further, quantitative characteristics tend to be influenced heavily by environmental effects as well. So we have an interplay between an organism's genes and the way that the environment affects that organism and impacts those genes. We also use the term discontinuous characteristics for qualitative traits, meaning there is not a continuous spectrum. There is boom, one state, or boom, the other state. Discontinuous. Whereas quantitative traits are referred to as continuous because you have this sloping spectrum from one extreme all the way continuous with representative phenotypes all along the way to the other extreme. More examples of quantitative traits would be blood pressure, weight, the rate of growth. In cows, for example, we'll come back to this example later, milk production. You don't have cows that make tons of milk and cows that make a little milk. Some cows make more milk than others, and we have a varying spectrum, a continuum of different cows producing different levels of milk. The many different DNA inputs, or we can also use the word genes, the many different genes that contribute to a quantitative trait are referred to as QTLs, quantitative trait loci. Loci referring to locations along the DNA, that's where the genes are. Traits referring to the fact that these locations of DNA impact a physical trait. And quantitative because we're not talking about qualitative traits, we're talking about quantitative continuous traits. So quantitative trait loci, the genes or DNA regions that contribute to a quantitative characteristic. And here's a perfect example. This is from a plant, not a pea plant. I believe this is a tobacco plant, but I'm not really sure anymore. And the color code tells you which trait is being influenced, and these are chromosomes along the plant genome. 3 different chromosomes are shown here, a subset of the whole genome. And so each one of these ovals represents a DNA region on that chromosome. So if we're interested, for example, to see the seed weight, what regions along the DNA, what regions in the genome contribute to the weight of the seeds of this plant? Well, we've got overlapping DNA regions here. Let me get my trusty pen. We've got DNA regions. I keep picking highlighter, I apologize. We have a DNA region here that contributes to seed weight. Seed weight. We have a DNA region here, right, that corresponds to these regions. And we have a DNA region here. So 3 regions of DNA on this first chromosome. But we also have a DNA region here. Now these are DNA regions. Usually they're genes, but they can also be DNA regions that contain numerous different genes for the same trait. They can even be other things, which we'll talk about in just a second. But here we have 4 different QTLs, 4 different loci that contribute to a quantitative trait such as seed weight or seed number. All right, so most QTLs are genes, are true protein coding genes, but other DNA sequences can contribute to a trait, and they would be a QTL. All we're talking about here are DNA regions that contribute to a trait. So what could be an example of a DNA region, a DNA sequence, that contributes to a gene, that trait, but is not a gene? Well, remember, in the last lecture we talked about regulation of genes. We talked about regulatory regions, such as operons and operators and promoters. We talked about regions of DNA that served as binding sites for other proteins, such as the LAC-I protein bound to the operator of the LAC operon. So sequences of DNA can play roles in gene expression. Sequences of DNA can play roles in phenotype, but not themselves necessarily code protein. A mutation in one of those regulatory regions that causes a regulatory protein to no longer be able to bind is certainly going to affect gene expression. It's certainly going to affect the outcome of the trait. But the mutation occurred in a region of DNA that itself was not protein coding. So these regulatory regions are not by definition genes because they don't encode proteins, but they certainly are DNA regions, and they're certainly contributing to a trait, and so they most certainly would be defined as QTLs. That's why we use the term QTLs and not genes, because other DNA regions above and beyond genes can contribute to quantitative traits. Now, we made the point in the earlier slide that sometimes traits are influenced both by the DNA, the inherited genetic information, as well as the environment. Any trait that does have an environmental component is called multifactorial, many factors contributing to the trait. And today we will be discussing, studying, and interpreting quantitative traits, QTLs, and the environmental effect on these traits. So we will be talking about multifactorial traits as well. That's what we're going over today. That's what quantitative genetics is. It's the study of traits that are continuous, that are quantitative, and that have many different DNA inputs on them, as well as environmental components as well. All right, so let's put a finer touch on the relationship between DNA information and the appearance of the organism, between genotype and phenotype. Everything we've discussed thus far, as far as it's gone in genetics in this course, has been very, very simple. There's one genotype, which equals one phenotype. We have discontinuous qualitative traits. That's what we've been talking about. And yes, we had some curveballs in all that. Dominant and recessive epistasis, codominance, incomplete penetrance, all of that kind of confused the simplistic Mendelian genetics that we had gotten comfortable with. But still, in the end, a single genotype, once we determined all the things that played into it, whether it was epistasis or codominance, once we knew the rules we were dealing with, a single genotype could be relied upon to give a very predictable and qualitative phenotype. A yellow dog. You don't need to measure the color of the dog, and even if epistasis is involved, once you know those rules, if I give you a genotype for dog coat color, you can tell me the color of the dog. With quantitative traits, it's not like that. It's not nearly as simple. First of all, we have polygenic traits. So multiple genotypes can give the same phenotype because all these different genes have their say in the trait. Therefore, seeing the phenotype doesn't allow you to reverse infer the genotype either, because multiple genotypes give the same phenotypes. Now, you add in an environmental component for multifactorial traits, and you have one single genotype can give multiple phenotypes. So everything is confused. Many different genotypes can give one phenotype, and one genotype can give many different phenotypes. In the case of multifactorial traits, knowing the genotype doesn't allow you to predict the phenotype if you don't know information about the environmental component at all. Let's go back to plant height. But let's consider plant height as a continuous characteristic. This is not in pea plants now. This is in a more advanced plant species. So plant height is going to be a continuous trait. It's going to be a quantitative trait. It's going to be a very simple continuous trait governed by only 3 loci, so only 3 genes are going to play a role in this trait. And each gene only has 2 alleles, so believe it or not, as confusing as it's about to get, this is a very, very simple case of quantitative genetics. So 3 genes all contribute to plant height. Each gene has 2 and only 2 alleles. We'll refer to them as plus and minus. Every single plant, regardless of its genotype, starts off at a height of 10 centimeters. And then, for every plus allele that plant has at any of those 3 loci, we add another centimeter of height to the organism. This is a very traditional kind of scenario. We have 3 genes, but we have a diploid organism, so there are 6 alleles there, right? The organism can be homozygotic plus, heterozygotic plus minus, or homozygotic negative. All right. So, if we have a plant that's homozygotic plus at the A gene, that means it's A plus. That plant is going to grow 2 additional centimeters because every single plus allele gives you an extra centimeter. A plant that's heterozygotic at A, 1 plus allele and 1 minus, is only going to grow 1 centimeter. The minus allele here has no effect at all. And the plant that is homozygotic negative at A won't grow any higher than those 10 centimeters because they don't have any plus alleles to add any height. But apply these rules to all 3 loci. There's an A gene, a B gene, and a C gene, all functioning the same way. Mathematically, we have 3 genes. We have 2 alleles per gene. That winds up being 27 different possible genotypes. And what we mean is we're considering all 3 genes. So A plus, B plus, B plus, C plus, C plus is a genotype. A plus A minus, B plus C minus is a genotype. A plus B plus B minus, C minus is a genotype. There's 27 different combinations of those given just positive and negative alleles. But even though there are 27 different genotypes, there are only 7 different possible phenotypes. Why? How could that be? Well, imagine I told you that there was a plant that was 14 centimeters tall. Okay? 14 centimeters tall. Well, you start off 10 centimeters tall, and you gain another centimeter with each positive allele. So you know from this that there are 4 positive alleles in this plant. That's how you got the 4 extra centimeters on top of the 10. But you don't know which of the alleles are positive, because any 4 positive alleles are going to give you an extra 4 centimeters. So any of these genotypes, as I've listed them up here, and there are more as well, any of these gives you a 14 centimeter plant, because these genotypes have 4 positive alleles. In fact, any of these genotypes gives you a 4 centimeter plant. Any of these genotypes gives you a 13 centimeter plant. Any of these give you a 12, and so on. There's only one genotype that keeps you at 10 centimeters, and that's when they're all negative. And there's only one genotype that gives you a 16 centimeter plant, and that's when they're all positive. But for all the phenotypes in between, there are multiple different genotypes that can give you that phenotype. Multiple different genotypes giving you the exact same phenotype. A characteristic of quantitative traits. Now again, this is a very, very simple example for quantitative traits, and this example is complex. If we start adding environmental components, if we start adding one set of alleles or one gene affecting another, things get infinitely more complex. So the example I just gave you was not multifactorial. In this case, the environment did not play a role. Environment played no role in that phenotype of plant height. It's quite unrealistic to think that. No matter how genetically determined a trait might be, it's extremely rare for any trait to be completely isolated from environmental effects. The environment plays some role in almost all traits. And the environmental effects on a trait or on a genotype cause some variation in the phenotype. So cause some of that continuous spread along the spectrum of the phenotypes. Let's consider the same example now. The same plants, 3 loci, 6 alleles, start off at 10 centimeters, etc. As I told you, the only way that a 16 centimeter plant could exist is if it had all positive alleles. It was homozygotic positive at all 3 genes. But now imagine that same plant is malnourished. It's not getting enough nitrogen. It's not getting enough sun. It's not getting enough water. It's not getting the environment it needs to thrive. I'll tell you right now, that plant is not going to grow to 16 centimeters, just due to a general lack of energy that it needed to grow. So environment does play a role. Most of the time, the phenotypic ranges of different genotypes overlap. They overlap due to environmental effects, making it very, very difficult to reverse infer the genotype from looking at the phenotype. I'll give you an example. Here's an organism on this spectrum of height. It's one of these plants. Here's the organism, and this is its height. It's on the short end of the spectrum, but it's not at one extreme and certainly not at the tall extreme. What's the genotype of this plant? Is this plant a nearly average heighted homozygous dominant plant? So it is falling here on the height spectrum because it's landing here on the spread of homozygotic organisms? Or is this a plant that's quite short on the heterozygotic spectrum? Because we see heterozygotes of the very same height. They're just a little bit shorter than average for that genotype. The short answer to this question is you don't know. Without looking at the DNA of the organism, you don't know. You cannot infer genotype from phenotype alone, not when you're dealing with quantitative characteristics. This organism can be either homozygotic dominant or heterozygotic, because we see individuals of that height in both of those genotypic classes. The phenotypes are overlapping with those 2 different genotypes. So we can't assign a genotype based on phenotype alone anymore, not when we're dealing with quantitative traits. At least we can't without some really, really heavy-duty statistics. We're going to talk about the power the statistics gives us in quantitative genetics, but there's going to be very, very little math in this lecture. Very, very little math you'll be responsible for, and we're not going to go through the statistics itself. We'll just more talk about what the statistics tells us when it's done properly. So put all those statistical fears at bay. So what are the different types of quantitative characteristics? In theory, a continuous characteristic can have any value between its two extremes. For height, there are extremes in the human species. There is a shortest person who ever lived and a tallest person who ever lived. Believe it or not, the smallest human being to ever live was 21.5 inches tall. That's not even 2 feet. And this individual lived into adulthood. Tallest person on record is 8 feet 11 inches. Man, oh man, can you imagine? Almost 9 feet tall. So those we can consider to be the extremes of height on the human spectrum. But there's an almost infinite amount of possibilities in between. I'm 5'9", technically. I round up just a touch there, but grant me that. My brother-in-law is also 5'9". Our driver's licenses say the same height on them. We measure ourselves to the same height. However, if he and I stand heel to heel, we're not the same height. He's a fraction of an inch taller than I am. I would guess somewhere in the neighborhood of a 1/4 inch taller. But we each call ourselves 5'9", and we're very, very close to 5'9", both of us. But we're not the same height. We still vary. So even with continuous characteristics, we have to, for the sake of dealing with the trait, we have to group ourselves into what we call these phenotypic bins, just for convenience. So 5'9" is a phenotypic bin. Anybody who's near that height calls themselves that phenotype, but it's not exactly true. I'm probably something like 5'8.8" inches tall. And if you have a really, really, really accurate ruler, I'm probably something like 5'8.87" inches tall. And my brother-in-law is probably something like 5'9.12" inches tall, if we were measured precisely. So he is a touch taller than I am. We are different in our height phenotype. We are in two different places on that continuum of height. Yet we bin each other and group each other to 5'9", for example. I round up and he rounds down. But there's another category of continuous characteristics that are not subject to rounding, and they are called meristic characteristics. Meristic characteristics. They're quantitative, but they're not continuous the way that height is. See, meristic traits or characteristics are measured in whole numbers. They can't be measured in fractions the way that height can be, 5'8.87" inches tall. But they still vary continuously, these meristic characteristics. Perfect example, litter size in cats. Litter size in cats is a quantitative trait. It's governed by many, many QTLs, and it varies across a spectrum from cat to cat. But it can't be measured in fractions, even if it's measured precisely, because cats don't have 5.87 kittens or 5.75 kittens. Cats have kittens in whole numbers. Cats have 12 kittens, 7 kittens, 8 kittens. So it's measured in whole numbers, yet still a varying continuous trait. Meristic characteristics are quantitative, they're studied as such, but they can't be measured in fractions because they don't occur in fractions. Another example is seed number. Right? Plants vary in the number of seeds that they make, but you can count the seeds in whole number. Seeds are not born in fractions. Another unique type of quantitative characteristic, threshold characteristics. Threshold characteristics appear qualitative because it appears as though there are two states, but they really aren't. Threshold characteristics are quantitative. The tricky part for threshold characteristics is that, like the name suggests, there is some threshold after which the trait shows up phenotypically and can be observed. But prior to the threshold being reached, the trait does not emerge. And so it doesn't exist. This gives the illusion of a discontinuous qualitative trait, because it's either yes or no, trait or no trait. But what's going on behind the scenes is a quantitative approach to this threshold. Let's use an analogy first, and then we'll kind of put a final definition on that. Imagine there's a very, very heavy door. and you need to close this door. It's like a vault door. It's very, very heavy. It's on great hinges, but it's still heavy. So what do? Well, the door starts off being open. And when you're done doing what you're doing, the door is going to be closed. So it looks discontinuous. It looks like two states. Door open, door closed. But when you approached this door to shut it, you started pushing on the door. Nothing happened. You started pushing some more. Nothing happened. You pushed more and more and more, applying more and more strength. Nothing happened. You know that you are pushing on this door with a continuum or a spectrum of strength. But to the observer, it's door open, door closed. Now eventually, you've pushed and pushed and pushed more and more strength. Eventually the door swings shut. And it looks to me like you walked up to the door, nothing happened for a while, the door stayed open, and then you shut the door. Two states, discontinuous, qualitative. But what you know is that there was a threshold of strength that you reached. You pushed, nothing happened, you pushed, you kept pushing harder and harder and harder and harder, and then eventually you pushed hard enough and that door swung shut because you reached the threshold of power of strength needed to shut that door. This is how threshold characteristics work. Diseases, cancer is a good example, but diseases are usually threshold characteristics. There are two states, diseased and not diseased, or healthy, and so it looks discontinuous. It looks qualitative. Disease, no disease. But in fact, it is a quantitative trait with a spectrum of phenotypes that remain invisible, just like pushing on that door with more and more strength. These people on this end of the continuum are very, very healthy and very, very far away from disease. These people, on the average of the spectrum, are quite healthy and still quite away far from disease. These people on this end of the spectrum are still healthy, but they really are just a toe away from developing the disease. One more environmental or genetic insult, one more environmental or genetic situation gone awry, and these individuals will become diseased. So there is a spectrum. There's a spectrum from mega healthy to barely healthy. But what makes these threshold characteristics look qualitative is that if you were to meet these three individuals on the street, very healthy, or barely healthy, none of them have disease. And so all of them look the same phenotypically. But they're not. They're weakly pushing on the door, moderately pushing on the door, or pushing on the door just barely hard enough to make it swing. With just a touch more strength, we would flip to the next phenotype. So threshold characteristics are most assuredly quantitative, but they appear to the outside observer as quantitative because of their two states. For these types of traits, when enough genetic and environmental effects come together in the right configuration, the disease develops. But before that critical mass, before that threshold, the individual is disease-free. And all disease-free individuals look the same phenotypically, despite their differences on that continuum. Because threshold traits are multifactorial, and they are usually governed by many different genes, they are quantitative, most definitely. So now on to polygenic inheritance. When Mendel's work was rediscovered by botanists in about the year 1900, most biologists took a look at that work and said, you know, that's great for pea plants, and that's really interesting, but that's an obscure exception to the rule. When it comes to human beings, there's no way that's how it works. Because peas might be green or yellow, tall or short, qualitative in so many ways, but people are most definitely varying on a continuous spectrum. People are 5 foot 9.7 inches tall, 5 foot 9.9 inches tall, 5 foot 10.2 inches tall, and explain that with simple classic transmission genetics. Scientists said there's no way human traits can be controlled in this way. We're too continuous. We're too blended. But then, soon after, a scientist named Wilhelm Johansen, showed definitively and genetically that bean weight, the seed weight of bean plants, was controlled multifactorially by both genetics and the environment, giving variation on the trait. Soon after, George Yule, a mathematician, then proved mathematically how multiple genes working together according to Mendel's laws could give rise to continuous characteristics such as those seen in humans. There was no genetics done here. This scientist was purely a mathematician. Again, he was a theorist. So he didn't prove Mendel was right, but he did prove that Mendel's theory was possible if applied to human beings. It was proof of principle. About 10 years later, 15 years later, in the late 1910s, was a quantitative genetic characteristic demonstrated to be operating in a living organism. The work was on wheat, tobacco, and corn. Work on traits from these three plants showed that the inheritance of QTLs, of multiple genes, multiple DNA inputs, could result in continuous traits that varied on a wide spectrum. And here is that classic experiment. The trait being studied was the color of kernels in a wheat plant. So this was the definitive experiment that proved that, one, quantitative genetics was real and demonstrable, and two, that Mendel was not a nutcase, that his ideas did in fact hold true in all species on this planet. In wheat plants, the intensity of the red pigmentation in the kernels of those plants is governed by two unlinked genes, two genes on two separate chromosomes, two QTLs, each with two alleles. So very, very simple system. And these experiments were started very much like Mendel's own experiments. A true breeding, white kerneled plant was mated with a true breeding purple, or in other words, very, very dark red kerneled plant, and 100% of the progeny were red. So a trait that was in between the two. Not quite white, not quite purple, but red. Looked like partial dominance, like our white flowers and our red flowers giving pink flowers. Then they did the F1 cross, and if you remember from partial dominance, when we talked about that way back when, if we do the F1 cross and we expect everything before it to be true, we're crossing a heterozygote by a heterozygote, so we should get 25% purple, 50% pink again, and 25% white. And this is what they did get. 1/16 of the plants were purple, 1/16 were dark red, 6/16 were red, 4/16 were light red, 1/16 were white. Not at all what was expected. The real story behind this trait is that it works very much like the plant height example we used a little bit earlier in this lecture. There are dominant plus alleles that contribute red color to the trait, and there are negative minus recessive alleles that don't contribute color. So to be a purple kerneled plant, you need 4 positive alleles, and there's only one genotype that gives that. To be a white kerneled plant, you need no positive alleles, and there's only one genotype to give that. To be a dark red plant, you need 3 out of the 4 alleles to be positive. To be red, it's 2 out of the 4. To be light red, it's 1 out of the 4, etc. And these are the genotypes that give those traits. These are what we refer to as additive effects, with each allele that is positive independently contributing its own red color. So there's no masking here. There's no dominance, because 2 positive alleles gives you more color than 1. 2 loci, 4 alleles, 5 phenotypic classes, purple, dark red, red, light red, white. As the number of the quantitative trait loci contributing to the trait increases, the simpler way to say that is as the number of genes that play a role in a trait increases, so too does the possible combinations of alleles, so too does the possible number of phenotypes. You get enough phenotypes due to enough QTLs, take those phenotypes together, and what do you get? You get a trait that appears to vary on a spectrum. I'll show you what I mean. One locus. Now these are additive effects. Keep that in mind. These are additive effects. So each allele that is dominant or positive contributes to the trait. If you have additive effects and you're dealing with one gene and 2 alleles, you have 3 possible states. Homozygotic negative on one end of the spectrum, heterozygotic in the middle, blended, partial inheritance, partial dominance, and homozygotic positive on the other end of the spectrum. Two loci with two alleles each. We get what we have here. Both ends of the spectrum, purple and white, and then dark red, red, light red, exactly as we're seeing here. 5 phenotypic classes. Let's move up to 5 loci. 5 loci gives us many different phenotypic classes, from one end of the spectrum all the way to the other, with another 1, 2, 3, 4, 5, 6, 7, 8, 9 different phenotypic classes in between. Many, many loci. Well, the more loci, the more phenotypic classes. The more phenotypic classes, the more you can bin those classes from one end of the spectrum to the other, with many, many, many different, barely varying from one another phenotypes in between. This is a quantitative trait. This is the hallmark of a quantitative trait, and it is due to nothing more than many loci contributing to a trait additively, where each positive allele influences the trait, and there's no true dominance or recessivity. So enough phenotypes taken together appear to vary continuously on a spectrum. And how do you get so many phenotypes? With many, many loci contributing to the trait. So this is where quantitative traits come from, whether it's human height, plant kernel color, or anything else. It simply comes from multiple loci with alleles contributing additively to the trait. Now imagine that environment also played a role here. It didn't play a role for these red plants, but imagine that it did. We would further increase the complexity of the trait, making the link between the genetics, the genotypes, and the trait even harder to sort out. But okay, let's go back to kernel color in wheat. Let's tie it all together. Let's show you how we get a varying quantitative trait, but by bridging it with Mendelian genetics using nothing more than Mendel's laws of transmission. 2 dominant alleles, or 2 plus alleles, are going to give wheat kernels a red color. We have, remember, 2 genes, 4 possible alleles. Any 2 of those 4 possible alleles being dominant or plus is going to give us a red kerneled plant. So these are the genotypes that give us red kernels. These are the only possible genotypes that give us red kernels. Remember, this is what we started with, was a red kerneled plant. We started with a purple true breeding plant, so the only way you can be a purple true breeding plant is if you have positive alleles for all 4 loci, all 4 regions. This was your true breeding purple plant, mated with a true breeding white plant. Well, there's only one genotype that allows you to be white in this case, and that is no positive alleles whatsoever. So this was the first cross that was done for this experiment. And it's pretty easy to see what we're going to wind up with. We're going to wind up with heterozygosity at both of these genes, A plus A minus, B plus B minus. And if we follow our rules of additive alleles, 2 out of the 4 alleles are positive. That's our red plants. So that was what we got in the F1. Now we're going to do the F1 mating. So we're going to take one of those red plants and mate it with another. Believe it or not, this is a typical dihybrid cross. You could set these up as Punnett squares and see what's going to happen. Getting the heterozygote condition back from this cross is 1/2 for the A's, 1/2 for the B's. You can do the Punnett square if you don't believe me. So 1/2 times 1/2 is 1/4. So 1/4 of these plants is going to be red due to having this particular genotype. The probability of getting red plants that have this genotype, because these plants are also red, right? To be red, all you need are 2 out of the 4 alleles being positive, is 1/4 times 1/4, which is 1/16. The probability of being red with this genotype, that's our last genotype of the bunch, is also 1/16. So what is the probability of being red, having this genotype, or this genotype, or, that's an or statement, the sum rule of probability, we must add our probabilities. You can be red with this genotype, or this one, or this one. And so 1/4 plus 1/16 plus 1/16 is 6/16. 6/16 of these plants are going to be red. And look, 6/16 of these plants were red. So we've done nothing but a dihybrid cross. We've done nothing but follow Mendel's rules. We've done nothing but apply our probability rules, which we started with over a month ago in this course. But we can explain the ratio for a quantitative trait. We can see how we got 6/16 red plants doing nothing more than a typical Mendelian dihybrid cross. The only difference was the new additive effect of these alleles. These alleles contribute additively. So now we have multiple genotypes giving the same exact phenotype because of that additive characteristic. You can do this for the others too. You can first isolate all of the different genotypes that would give you a dark red plant, and then compute the ratios for each of those genotypes and add them together. You'll get 4/16. You can do the same for the light red if you'd like. Based on all of this, hopefully, at this point, you're comfortable with two broad concepts. One, many traits, especially the more complex traits in more complex organisms, are governed by multiple genetic inputs, multiple regions of DNA, made up largely of protein-coding genes, but also including other DNA sequences, and we collectively refer to these as quantitative trait loci, QTLs. Because we have many traits being controlled by many different genes, that makes those traits vary almost infinitely along a broad spectrum. And this is the definition of quantitative traits. Remember, many different loci, each with alleles adding additively, gives us many phenotypic classes. We saw that. We demonstrated that. 2. In addition to these multi-DNA inputs, these QTLs, we also have to consider environment playing a role in the expression of traits as well. Most traits are multifactorial, made up of a combination of genetic and environmental inputs. If we have any hope whatsoever of predicting the phenotype of an organism from its genotype, we must determine how much of that given trait is impacted by the genetics and how much is due to the environment. If we don't know the proportion of the trait that's controlled by genetics versus the portion of the trait controlled by environment, we can never hope to predict phenotype from genotype. Without the information of the environmental impact on a trait, we're wasting our time as geneticists. We made the point much earlier in the course that geneticists are in the business of prediction. That's how we make our bread and butter. That's how we make our money. You give us parents, and we can predict the ratios of the offspring. Now you're telling us that environment impacts the gene? The environment impacts the trait? Well, if I have any hope of keeping my business going and doing my predictions, I have to know how does the environment affect the trait? Where does the environment affect the trait? In which way does the environment affect the trait? And how much is that effect there? What is the effect, the proportion of the effect that the environment has? Without knowing that, there's no certainty at all whether there's a genetic component to the trait to begin with. So let's talk now about this idea of heritability. In its most simplest conceptual state, the idea of heritability, the definition of heritability, is the proportion of the total phenotypic variation that is due to genetic differences. Okay, so how much an organism differs in its phenotype from one extreme to the other, that idea of the continuum or spectrum of phenotype, how much of that is due to genetics is heritability. Let's go back to cows and their milk. A farmer notices that some of his cows are producing much more milk than the others. The farmer is very happy. He's a dairy farmer. Milk is money, so this is good. If this difference in cow milk production is due to genetics, the farmer knows exactly what he wants to do. He can get the male cows and selectively breed them with only the female cows that are making the most milk, creating a next generation of cows that make more milk too. He can selectively breed only the cows that are making more milk so that his next generation will be lots of milk producing cows as well. But if the difference is environmental, the farmer knows exactly what he should do. He should pay special attention to the environment that the higher milk-producing cows are in. Do they spend more time indoors? Do they spend more time out of doors? Are they preferentially eating different types of feed? Are they getting to a different patch of grass? What are they doing differently? What are they getting from their environment differently that the less milk-producing cows are not doing? And then the farmer can simply take his lower milk-producing cows and move them into an environment that is much more like that of the high-producing cow. You see, the farmer wants to get his low-producing cows to be like his high-producing cows. But his strategy to do so changes dramatically based on whether or not this difference in milk production is due to genetics or due to environment. By knowing and measuring the genetic component of this milk-producing trait, and that is the heritability, by being able to compute and calculate how much is the milk production trait governed by genes, and how much is it at the mercy of the environment, only with that information can the farmer understand and then manipulate that trait. And the first step to understanding this is measuring it. And we measure these things using a frequency distribution. We measure these things by binning these animals. We preset some limit to how we want to bin these cows together, and then we do so. And we look to see what the distribution of the cows are on the farm. I have no idea how much milk a normal average dairy cow makes in a day. But let's say, for argument's sake, it's 3 gallons. So if that's average, then we have a 3-gallon milk-producing bin. And then we have 3.1 gallons, 3.2 gallons, 3.3 gallons, 3.4, all the way perhaps to 4 gallons. And then 2.9, 2.8, 2.7, all the way down to 2. And so we have a spectrum of milk production from our lowest extreme to our highest extreme. And we just go to the farm and we count how many cows are each in each bin. How many cows make 2.4 gallons of milk? How many cows make 3.7 gallons of milk? And we do a frequency distribution. We determine the number of cows in each phenotypic class. That's the first step. And then we apply a lot of statistics and we measure the phenotypic variance. So we obtain a random sample of cows, right? Those are the cows on the farm, making up our larger population. We measure it for the trait we're studying. We do this frequency distribution, everything I just showed you in the bar graph. We then calculate the average, or the mean, of milk production. In that bar graph, the average was going to be 3 gallons. And we also calculate the variance. The variance is a modified version of the standard deviation, which you've probably encountered in other courses. A variance is a measure of how much these cows spread on either side of the average. This variance is our phenotypic variance. The spread of the phenotype around the average, the deviation of the phenotype around that average to one extreme and then the other. So the phenotypic variance is a measure of how diverse are the phenotypes. How different do they go? How much variation is there? How far is the spread? How broad is the spectrum? That's our phenotypic variance. Some of the phenotypic variance will be due to genetics, of course, due to the differential inheritance of alleles, due to our QTLs. And we break it down and say this is the genetic variance. The genetic variance is the proportion of the phenotypic variance that's due to genes. And some of the phenotypic variance, some of the spread in that continuum, will be due to the environment. Can you guess what this one's called? You got it. That's the environmental variance. So the environmental variance, together with the genetic variance, explains the phenotypic variance. In other words, how much these cows are different than each other due to the environment, as well as how much these cows are different from each other due to their genetics, makes up the sum total of how much they're different from each other. Period. Now, environmental variance is a catch-all. Yes, it includes the environmental differences, but it includes everything that isn't genetic. So any variance in the phenotype that can't be explained by genetics is thrown into the environmental variance. In other words, any variation in the phenotype that cannot be inherited is deemed environmental. That's all pretty straightforward. But sometimes the genetics of a trait depend on the environment. Remember that Arctic hare we talked about way back when with incomplete penetrance? It turns black when cold reared at its tips, at its coldest points, but it stays all white when it's warm, when it's raised in the warmth. This is an environmental specific effect, right? Because the temperature plays a role on the phenotype. What that bunny looks like is due largely to its environment. But it's also due to genetics, because the right genes need to be in place to allow that environment-specific effect to occur. There are genes that make that bunny's fur change color due to temperature. So this is an environment-specific, genetically based effect. We call this genetic environment interaction variance. This is the amount of phenotypic variance that's due to an interplay of certain genes and the environment. Not completely environmental, because it's genetically based. Not completely genetic, because the environment serves as a trigger. So it's genetic environment interaction variance. Here's another example. Imagine a plant, and there is such a plant. When the plant is dry, so when it's raised in more arid conditions, if it has a homozygous dominant genotype big A, we have a plant that has an average weight of 12 grams. And the homozygotic recessive plant, in an arid condition, weighs less, about 10 grams. But when the environment of this plant is wet and there's an abundance of water, the homozygotic recessive plant weighs about 24 grams. It grows much bigger because it's got more water. And the homozygotic dominant plant weighs an average of 20 grams. They switched. So when it's dry, the homozygotic dominant is heavier than the homozygotic recessive. When it's wet, the homozygotic dominant is lighter than the homozygotic recessive. The environment, the abundance of water, flipped the phenotypic pattern. It flipped the phenotypic pattern, making the tall guy short, or the heavy guy lighter, and the light guy heavier. That is a genetic environment interaction variance. There have to be genes for that, because we're dealing with genotypes, but there's an environmental component too. The weight of the plant is not due solely to the environment. The weight of the plant is not due solely to the genes. The weight of the plant is due to an interaction between those two factors. So we can't split the phenotypic variance between the genes and the environment. We can't do that. We can't say the variance in the phenotype is due this much to the genes and this much to the environment, and they're completely independent, because they're not independent. The two are entangled. The environment influences the phenotype. The genetics influences the phenotype, but they must work together to do so. So that gets its own unique class of variance. Back to traditional genetic variance, though. What are the different components of genetic variance? Well, we divide genetic variance into different types of genetic effects, and we're familiar with these already. We're just giving them new names in this kind of new framework of variance. Additive genetic variance is what we've been talking about all this lecture. It's when alleles contribute independently and additively to a trait, such as the height example we started with 1 centimeter for each positive allele, such as wheat red kernel color, where each positive allele makes the kernel a little bit redder. These are additive genetic effects. They contribute to additive genetic variance. Dominant alleles contribute each independently to increase the trait additively. You're twice as red with 2 dominant alleles as you are with 1. Each positive allele causes the plant to grow 1 centimeter higher, regardless of whether you're dominant or recessive. In other words, a homozygotic plus plant grows 2 more centimeters. A heterozygotic plus plant grows 1 more centimeters. Additive effects. Additive effects. Then we have dominance genetic variance. Dominance genetic variance is our classic Mendelian rules applied to this kind of framework. Dominant genetic variance is when there are 2 alleles, one is dominant and one is recessive, and the dominant allele trumps the other. In other words, the alleles are not additive. There is no difference. When you're dealing with dominance genetic variance, there is no difference between homozygotic dominant and heterozygotic. Either you have the genetic information for the dominant trait, or you don't. No difference between homozygotic dominant and heterozygotic. That's dominant genetic variance. And then genic interaction variance. This refers to the idea of epistasis. Separate independent genes interacting with one another, influencing a single trait. The example of coat color in dogs is a perfect one. Genic interaction variance. Variance in the genotype, golden dog, black dog, due to the interaction of different genes. E is epistatic and controlling B. So it's at this point in the lecture that I can fully sympathize if you're looking at the screen in a very odd way, perhaps with a crooked eyebrow, and you're saying to yourself, shoot me now, when will he stop speaking, what the hell are we talking about? So let's take a deep breath and kind of back up, zoom out a little bit, and get an idea of the big picture once again. All we're talking about here, all we're doing, is trying to account for everything that contributes to the phenotype. People come in different heights. That's the variance of the phenotype. People come in different heights. The phenotype for people height varies. And we're trying to consider why there's that variance. Why are people different in height? And so we come to the conclusion that some of that variance in phenotype, some of that variance in height, is due to the different genes we're walking around with. That's the genetic variance. And some of the variance in height between people is environmental. And that's the environmental variance. So the genetic variance plus the environmental variance makes up the total phenotypic variance. In other words, it accounts for and explains all of why we are different in height. But we're breaking the genetic part down, and we're saying it's not just enough to say that there's genetic variance. There's actually differences in height in people due to genes for 3 different reasons. Some of those genes work additively, where each positive allele contributes to height, and so we call that additive genetic variance. Some of those genes that affect people height follow the classic Mendelian laws of dominance. You're dominant, you have one dominant allele, you show that contribution. You have 2 dominant alleles, I don't care, you only need 1. You show the same contribution. They're not additive, they're classic dominance, and we call that dominance genetic variance. And then some of those genes that affect height in people are subject to epistasis. They're subject to control by other genes. In other words, multiple genes are interacting, masking and unmasking each other for people height. And we call that genic interaction variance. So genic interaction variance plus dominance genetic variance plus additive genetic variance makes up the sum total of the genetic variance in height. Add to that the environmental effects, the environmental variance, and you have accounted for everything that contributes to height in people. That's all we're saying. Nothing more. We're breaking down the differences in phenotype into each of its smallest components. All right. So let's move on then to types of heritability. Each of the components we just defined and described come together when brought together to explain all of that phenotypic variance observed in a population for any one point, any one trait. That was the point of all of this. That's what we were trying to do. Account for, and I mean account for, like an accountant. Put a number to, put a proportion to, account for all of the different inputs that affect a trait. This also often allows us to kind of separate, dissect, parse out how much of a phenotype is due to genetics, obviously, and how much is due to something else, and by something else we usually mean the environment. The idea of a broad sense heritability represents the proportion of the phenotypic variance that's due to genetics. Mathematically, a broad sense heritability can be measured using statistics, which we will not do. It can be measured and computed, it can be calculated, and it can range from 0 to 1. When you have a 0 broad sense heritability, that means your genetic variance is 0. That means you have no genetic component at all, and all of the variance of your trait is due to the environment. Perfect example is the team that you follow for a given sport. I'm a die-hard Bruins fan. I'm a big hockey fan. I love the Boston Bruins. I always have. Since the first day I started following hockey, I was a Bruins fan. Even when I was a New Yorker, I was a Bruins fan. So I'm finally made it here into home country. There is a 0 broad sense heritability for being a Bruins fan. Now, in my family, it seems to be an inherited trait. My older daughter likes hockey. She's beginning to be a Bruins fan. My younger daughter, following me and her older sibling, also seems to be a Bruins fan. So my children seem to have inherited that trait from me. My wife is not a hockey fan at all, so there is a phenotypic variance in our family. There is a difference. We differ from one another. I'm a die-hard fan. My older daughter likes it quite a bit, but not as much as me. My younger daughter is just getting interested now, but I wouldn't call her a big hockey fan. And so there's a spectrum of fandom in our family. But none of that is due to genetics. It's entirely environmental. Even if it appears to be inherited in the family, it has a broad sense heritability of 0, meaning there's no genetic component at all. A broad sense heritability of 1 means that it is all genetics, and there's no environmental component at all. In other words, 100% of the phenotypic variance is explained and accounted for by the genetic variance. So tongue curling is a good example here. Tongue curling follows a pretty similar pattern. My wife can do it, my kids can do it, and I can't. Very similar to the inheritance pattern of the Bruins fans. However, this is not environmentally influenced at all. It is entirely genetically based by a single gene. And so it has a broad sense heritability of 1. Traits with a broad sense heritability of 0.8 mean that, and this is a loose translation, but it's certainly sufficient for this course at this level, a broad sense heritability of 0.8 loosely means that 80% of the trait of the phenotypic variance is due to the genetics, meaning 20% is due to the environment. A broad sense heritability of 0.3 means 30% of the inheritance of the trait, or the differences in phenotype, are due to genes, and 70% is impacted by the environment. So any number between 0 and 1 shows you that both genetics and environment play a role, but also give you some idea of the weight that genetics and environment has on the trait, with numbers closer to 0 meaning more environmental, less genetic, and numbers closer to 1 meaning more genetic and less environmental. But we also have a narrow sense heritability, and the narrow sense heritability is different than the broad sense. The narrow sense heritability is still the proportion of a phenotypic variance, but now it's the proportion of the phenotypic variance that can be accounted and explained solely from additive genetic variance. In other words, solely from genes whose positive alleles contribute individually to the trait, like those plants growing a centimeter taller or those kernels being a little bit redder. Narrow sense heritability is extremely important for breeding. It's the narrow sense heritability that dairy farmer wants to know. Because when you know the narrow sense heritability, you can predict and breed traits in and out. Essentially, the narrow sense heritability is a measure for how likely offspring will resemble its parents. If the narrow sense heritability is high, what that really means is that most of the phenotypic variance is due to not only genetics, but it's due to additive alleles contributing equally and independently to the trait. When you have high narrow sense heritability, the correlation between the average parental phenotype and the average offspring phenotype is extremely high. That's the prediction that we're talking about. In other words, parent cows that make a lot of milk, or I should say mother cows that make a lot of milk, will give birth to baby cows that make a lot of milk. That's what a high narrow sense heritability tells you. If alleles are additive, Tall parents have lots of positive alleles, and they will pass those positive alleles to their children, making them tall. This is different from dominance genetic variance. See, because dominance genetic variance, two parents can come together if they're tall and have a short kid. Heterozygote crossed with heterozygote have 20% homozygotic recessive. So there's some predictive value there. But you can be a tall mom and a tall dad and give birth to a short kid with dominance genetic variance. If narrow sense heritability is high, that means additive genetic variance is high. That means a tall parent is tall because they have two positive alleles. A tall mother is tall because she has two positive alleles. So if two tall parents come together and mate, they're going to have kids with nothing but two positive alleles, and all those kids will be tall. Additive genetic variance is the best predictor for parents and offspring having the same phenotypes. When additive genetic variance is 0.5, we have less of a correlation between the parent's phenotype and the children's. And when additive genetic variance with the narrow sense heritability is 0, there is no correlation. There is no correlation. Tall parents can have short kids or tall kids. Short parents can have short kids or tall kids. There's no correlation when there's low additive genetic variance, when there's low narrow sense heritability. So if we have high narrow sense heritability, we have high variance explained by additive genetics, we're not worrying about dominance or recessivity because each allele contributes independently. We're not worried about epistasis because all of that variance is in the additive genetic component. We're not worried about environmental effects because that number also means that the environment's playing a low role. We're saying most of our trait is due to additive, independently contributing alleles. That's narrow sense heritability. So that's all great. That's all what heritability can tell us. That's all of the information we can get from heritability. But let's also talk about the limitations of heritability. If there is high heritability, and we can prove that there's high heritability, we can predict phenotype using nothing but genotype. Why? Well, if there's high heritability, that means there's a low environmental impact on the trait. And if the environment is playing a very small role, then most of the role is genetics, and geneticists can predict phenotypes when genetics are in play. That's what geneticists can do. That's what geneticists want to do. High heritability also tells us that an organism should or will respond to selection, like our dairy cows. We can breed in and breed out traits that we want to if those traits are genetically controlled. We can't breed traits in or out of a species if those traits are environmentally controlled. To change the phenotype there, we would need to change the environment, not the genes. But there's a lot that heritability can't tell us. First off, individuals do not have high heritability. Populations or species do. Heritability values represent the proportion of variance in a phenotype that's due to genetics in a population of individuals. I'll give you an example. If, for argument's sake, the weight of human beings has a genetic heritability value of 0.6, then we can say that 60% of the variance in human weight between people is genetically based, and 40% of the variance is environmentally based. That's what that tells us. That's a value for the population. It does not mean that 60% of my weight is due to my genes, and 40% of my weight is due to what I choose to eat. That's not what it means. Heritability does not apply to the individual. I don't have a phenotypic variance of weight. I have one weight. I step on the scale, I see a number. That's my weight. These values of heritability apply to the variance, why we're different from each other in the population, not how much of our individual trait is made up of genetics and environment. There is a difference. Also, when heritability is high, we've said that means that most of the variance in the phenotype is due to the environment, is due to genetics, and the environmental input is considerably lower. But even when heritability is high, the environmental factors can still influence a trait. Imagine this. There's plenty of water in a field. The water's been in the field for a long time. It's been raining in that field for decades. Plants are getting plenty of water. All of their variation in height is due to their genetics. High heritability, low environmental component. Now, same field, but there's a drought. Rain stops. Water's not coming anymore. The environment, which was stable for a very long time, just changed. And guess what? All the plants are now much shorter. Did the environment affect the phenotype? You're damn right it did. The water went away, the plants got shorter. But heritability was high. Heritability said that the trait was not affected by the environment. Don't care. The change in this trait is purely environmental. Just because the environment wasn't affecting the trait before, and now it is, doesn't mean that the environmental component didn't play a role. The textbook says the absence of environmental variation in a characteristic, in other words, a high heritability, does not mean that the characteristic will not respond to environmental change. Don't ever think because a heritability value is high, the environment cannot and will not ever play a role in that trait. It just means that in this population, right now, as this population currently stands, if heritability is high, the differences we see between individuals of that population is due to genetics. That's all it means. Finally, heritability is population specific. You cannot apply heritability values from one population to another, even if those animals are in the same species. It doesn't tell us why different populations differ from one another. In other words, we can't use a heritability value to explain the inheritance of these stripes in these different species of tiger. This population of tiger has a heritability value for its population that explains the genetic influence of the inheritance of its coloration pattern. That value, it does not apply to this population of tiger or this population of tiger. Different populations that are isolated from one another and not interbreeding have different heritability values even for the same trait. Okay, so what can we do with all this information as breeders and as geneticists? Evolution boils down to genetic change within a population. That's all evolution is. The changing of the genes, the changing of the DNA content within a population of individuals. Some of these changes make some members of the population more fit. As a result, they live longer, they procreate more, and they get those genes into the next generation. By procreating more, they spread those fit alleles. They spread that superior DNA more efficiently to the next generation, and they drive the species forward. That is natural selection. Natural selection is the survival of the fittest. Those organisms with the best genes for the here and now have the most babies and get those genes into the next generation. This is one of the primary drivers of evolution. It allows species to adapt to their changing environment, because as conditions change, who is more fit and why changes. And always the most fit, most surviving members of the species have the most babies and get their superior DNA to the next generation. Always the individuals best suited for that current environment survive and procreate best. And so natural selection is always occurring in real time. But we also have artificial selection. Humans are great at this. It's a very, very real, though unnatural process. This is breeding. This is taking those cows that produce the most milk and only allowing them to mate, only allowing those genes to make it to the next generation of cows on the farm. This is nothing new. We've been doing this for thousands of years and explains almost all domesticated crops that we eat, domesticated livestock. It explains pets. It explains so much of what is part of our everyday lives right now. Different breeds of dogs are the direct result of artificial selection, taking only those dogs with the flattest faces and mating them with other flat-faced dogs, taking only dogs with the best, most discerning sense of smell and mating them with other well-smelling dogs, taking the strongest dogs, the dogs that listen the most, the largest dogs, the most fierce dogs, breeding those mothers with those fathers that show the strongest examples of those traits, getting those genes to the next generation. That is the manipulation of evolution. That is artificial selection. And artificial selection does not work without high heritability, meaning the trait is genetically governed, and more importantly, with high narrow sense heritability, meaning that the alleles that contribute to that trait do so independently, and so you'll have the highest chance of offspring looking like parent. So quite a bit in this lecture, a very different flavor of lecture than the ones we've been having up to this point. A little bit more kind of abstract, but much closer to home. This is the most cutting-edge genetics we're going to cover in this course. This is the most kind of contemporary genetics that we're going to cover, and it does the best at explaining why we are who we are and why we look the way we look, still by following Mendel's simple laws of dominance and recessivity. So what did we talk about today? Well, we started off defining quantitative traits. Quantitative traits are those traits that are governed by many different genes, many different loci, these quantitative trait loci. The many different DNA inputs, those are genes, but other things as well, that contribute to a quantitative trait are referred to as QTLs. A multifactorial trait is a trait that is influenced both by a genetic component as well as environmental, and indeed most traits, certainly in our species, are multifactorial. We define different types of quantitative traits. Meristic characteristics are quantitative, but they're measured in whole number, like seed number or litter size. Threshold characteristics, they look qualitative, but they aren't. Healthy, diseased, state 1, state 2. But if you get behind the scenes and you look at the trait, you see there's a broad spectrum of health, from very, very healthy to just a hair's breadth away from being diseased, and so threshold characteristics are most definitely quantitative. The greater number of QTLs, the greater number of phenotypic classes, the greater spread we get in those phenotypes, the more that trait looks quantitative, and we kind of went through a little thought experiment there with the different histograms. And then we switched gears and talked about heritability. The proportion of the entire variation of phenotype within a population of individuals that's due to genetic differences is the heritability. We talked about the differences in phenotypic variance and genetic variance. Phenotypic variance is all of the variance between individuals for that phenotype. The genes play a role in that, and that is genetic variance, but environment plays a role too. And so we have environmental variance and genetic variance accounting for the phenotypic variance. We define the broad sense heritability as being a numerical value representing the proportion of the phenotypic variance that's due to the genes. The closer you are to 1 in this value, the closer you are to saying all of the variance in the phenotype is due to genetics. But then there's narrow sense heritability. Here we narrow in our focus of heritability and look only at additive genetic variance. So narrow sense heritability is a value from 0 to 1 that represents the proportion of the phenotypic variance that comes solely from additive effects of individually contributing alleles, like the 1 centimeter higher for plants, or the little bit more red for kernel color. And finally, we discussed some of the limitations of heritability. Individuals don't have heritability. Heritability applies only to populations. When heritability is high, you shouldn't think that environmental factors don't play any role at all. The environment can still play a role, even if heritability is high, such as a drought affecting plant height. And heritability is population specific. Even members of the same species can't be thought to have the same heritability if they are part of different isolated populations. And we wrapped up the lecture talking a little bit about breeding, natural selection, evolution, artificial selection, and how that is all genetically based and genetically governed. So we're in the home stretch now. We have 2 more lectures left for this course. As we move on, we're going to have a 2-lecture series where we cover population genetics, the last big subfield of genetics as a whole. Again, that's going to be a single textbook chapter broken up into 2 lectures, and those will be the last 2 lectures of the course. But as always, thanks for watching.